Faucet. Our mission is to bridge the gap between scientific research and practical healthcare applications, ensuring that the latest advancements improve patient care, health outcomes, and public health. Biomedical knowledge translation involves turning cutting-edge biomedical research into tangible solutions for healthcare systems. At Actuate Biomedicine, we specialize in this process, ensuring that scientific breakthroughs reach those who need them most. We host our Learn to Earn sessions every Tuesday from 7 p.m. to 8 p.m. East African Time. Each cohort participates in an 8-week program designed to guide learners through the 7 stages of biomedical knowledge translation. The 7 stages include exploration, where we identify new research and innovations; evaluation, assessing the potential impact and feasibility; planning, developing strategies for implementation; development, creating practical applications and solutions; implementation, integrating these solutions into healthcare settings; another round of evaluation, monitoring and assessing effectiveness; and finally, distribution, ensuring wide access to the final products. Our learners gain in-depth knowledge and practical skills, empowering them to translate biomedical research into real-world applications. Through our program, we aim to foster a new generation of experts who will drive advancements in healthcare. Join us at Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Together, we can make a significant impact on healthcare and public health. Learn, earn, and contribute to a healthier future. Be a part of the change. Transform knowledge into action with Actuate Biomedicine. Welcome to Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Our mission is to bridge the gap between scientific research and practical healthcare applications, ensuring that the latest advancements improve patient care, health outcomes, and public health. Biomedical knowledge translation involves turning cutting-edge biomedical research into tangible solutions for healthcare systems. At Actuate Biomedicine, we specialize in this process, ensuring that scientific breakthroughs reach those who need them most. We host our Learn to Earn sessions every Tuesday from 7 p.m. to 8 p.m. East African Time. Each cohort participates in an 8-week program designed to guide learners through the 7 stages of biomedical knowledge translation. The 7 stages include exploration, where we identify new research and innovations; evaluation, assessing the potential impact and feasibility; planning, developing strategies for implementation; development, creating practical applications and solutions; implementation, integrating these solutions into healthcare settings; another round of evaluation, monitoring and assessing effectiveness; and finally, distribution, ensuring wide access to the final products. Our learners gain in-depth knowledge and practical skills, empowering them to translate biomedical research into real-world applications. Through our program, we aim to foster a new generation of experts who will drive advancements in healthcare. Join us at Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Together, we can make a significant impact on healthcare and public health. Learn, earn, and contribute to a healthier future. Be a part of the change. Transform knowledge into action with Actuate Biomedicine. Welcome to Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Our mission is to bridge the gap between scientific research and practical healthcare applications, ensuring that the latest advancements improve patient care, health outcomes, and public health. Biomedical knowledge translation involves turning cutting-edge biomedical research into tangible solutions for healthcare systems. At Actuate Biomedicine, we specialize in this process, ensuring that scientific breakthroughs reach those who need them most. We host our Learn to Earn sessions every Tuesday from 7 p.m. to 8 p.m. East African Time. Each cohort participates in an 8-week program designed to guide learners through the 7 stages of biomedical knowledge translation. The 7 stages include exploration, where we identify new research and innovations, evaluation, assessing the potential impact and feasibility, planning, developing strategies for implementation, development, creating practical applications and solutions, implementation, integrating these solutions into healthcare settings, another round of evaluation, monitoring and assessing effectiveness, and finally, distribution, ensuring wide access to the final products. Our learners gain in-depth knowledge and practical skills, empowering them to translate biomedical research into real-world applications. Through our program, we aim to foster a new generation of experts who will drive advancements in healthcare. Join us at Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Together, we can make a significant impact on healthcare and public health. Learn, earn, and contribute to a healthier future. Be a part of the change. Transform knowledge into action with Actuate Biomedicine. Welcome to Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Our mission is to bridge the gap between scientific research and practical healthcare applications, ensuring that the latest advancements improve patient care, health outcomes, and public health. Biomedical knowledge translation involves turning cutting-edge biomedical research into tangible solutions for healthcare systems. At Actuate Biomedicine, we specialize in this process, ensuring that scientific breakthroughs reach those who need them most. We host our Learn to Earn sessions every Tuesday from 7 p.m. to 8 p.m. East African Time. Each cohort participates in an 8-week program designed to guide learners through the 7 stages of biomedical knowledge translation. The 7 stages include exploration, where we identify new research and innovations, evaluation, assessing the potential impact and feasibility, planning, developing strategies for implementation, development, creating practical applications and solutions, implementation, integrating these solutions into healthcare settings, another round of evaluation, monitoring and assessing effectiveness, and finally, distribution, ensuring wide access to the final products. Our learners gain in-depth knowledge and practical skills, empowering them to translate biomedical research into real-world applications. Through our program, we aim to foster a new generation of experts who will drive advancements in healthcare. Join us at Actuate Biomedicine's Biomedical Knowledge Translation Faucet. Together, we can make a significant impact on healthcare and public health. Learn, earn, and contribute to a healthier future. Be a part of the change. Transform knowledge into action with Actuate Biomedicine.
Hello, good evening, everyone. Hope I'm audible enough.
Yes, go ahead.
Yeah,
Yes,
I
go
think
ahead.
we can start the meeting. Thank you so much, everyone, for joining today's meeting. Today, we are going to be focusing on quadratic funding in the prototyping stage of biomedical knowledge translation. We're going to have an overview of what the prototyping is in the biomedical knowledge translation and how quadratic funding is important. So thank you so much for joining. You can see our topic master has already arrived. So without further ado, we're going to have a meeting just follows.
I'm sorry, Grace, we can't hear you. I think there's something wrong with your audio.
You're breaking off, breaking.
Sorry for that. I hope I'm audible now.
Yes, what did you do?
Yeah, I was saying that from there we are going to have a session for question and answer. Then we're going to conclude our meeting. So let me welcome Damin to continue and give us the topic for today. Welcome, Damin.
Thank you, Grace. Good evening, everyone.
Good evening, Demi.
Thank you very much. I hope everyone is doing good. So today, without losing time, today we have a session about quadratic funding in the prototyping stage of biomedical knowledge translation. So we are going to check the introductions of this, and then we are going to see the meanings of quadratic fundings as we daily learn about this. And also we're going to check if prototypings in a biomedical knowledge translations, and you are going to see on how we are going to apply these quadratic findings in a prototyping. We'll have some of the case studies in the quadratic findings actions, and you are going to conclude. So without losing time, so let's go start now. In every day that we just involving about this landscape of biomedical research and innovation. The process of this translating about biomedical knowledge from the laboratories into the real world applications is usually the complex, and sometimes is all about the manifested. And the one promising approach is to facilitate easy translations into quadratic funding. That is all about the method that optimize resource allocations by leveraging this corrective intelligence and preferences of the community. And this article explores more about this application of quadratic fundings in the prototyping stage of biomedical knowledge translation that highlight its potential to democratize findings and accelerate the developing of impactful biomedical solutions. Let's go deep to the meanings of quadratic fundings. Quadratic fundings is a decentralized funding mechanism designed to allocate these resources more equitably and effectively based on the community support. The core ideas behind quadratic fundings is to match contributions from a larger pool of funds in a way that gives more weight to the number of contributors rather than the amount of contributed. And this approach amplifies the influence of smaller donations, ensuring that a project with its broad-based community support receive more funding. So this means that if a project receives a large number of small contributions, it will receive more funding than a project with a few large contributions, even if the total amount contributed in the same. This mechanism encourages widespread participations of the different communities, stakeholders, supporting more projects that resonate with the large portions of the community. So, what is the meaning of prototyping? What do you mean when we say the meaning of prototyping? Prototyping is just a critical phase in biomedical knowledge translations. And this always involves developing, testing, and refining versions of biomedical innovations, such as medical devices, diagnostic tools. This is about X-rays, of sonographic, ECG, for detecting any diagnostic symptoms, medical conditions, which talks about the therapeutic interventions. So this stage arouse researchers and concepts by identifying their potential issues in the community and gather essential feedback from the stakeholders, from different healthcare professionals and patients. So during this stage, we are secure adequate fundings is crucial to cover all costs associated with materials, testing, regulatories, issue for interactive development. So the addition about these models, such as grants and ventures capital, often come with limitations, including these issues of application processes, eligibility criteria, and the potential barriers. So, quadratic funding play a great role in transformatives about this stage. So let's see how can we apply this quadratic funding in prototyping. The first one is all about to democratizing access to funding. But how? Let's take quadratic funding. It's level up about the playing field by allowing a diverse group of stakeholders maybe to participate in setting funding processes. About researchers or patients or healthcare providers and general public health can all contribute to the project that they believe in. These democratizations of funding ensures that a broader range of ideas and innovations have the opportunity to be developed and tested and to be considered about these researchers, patients, healthcare providers. We all gather their ideas in order to innovate a problem in a community, to come with good measures to solve certain problems according to biomedical knowledge translations. And another applications, it was all about to encouraging collaborations and community engagement. As we always learn, this quadratic funding fosters a sense of community and collaborations among stakeholders. By how? This contribution to a certain project, individuals become more invested in their success and are more likely to provide about their variable feedback about a certain project and support throughout the prototyping process. And these collaborative ideas bring a good approach, which can lead to more robust and user-centered biomedical innovations in these healthcare services that are provided to the hospitals. And another, it is all about enhancing resources allocations efficiency. Quadratic funding in this part, we always say this is all about a mechanism that efficient allocates about these resources to projects with guidance community support. So this enough resourcing reduce the risk of funding being concentrated in a few high-profile projects. We are just while neglecting about these small potentially groundbreaking innovations. So this ensures that funds are distributed based on the collective preference and needs of the community. Another is all about the incentivizing broad-based support. Quadratic funding always use these formulas, and these formulas used by the project to seek support from a wide range of contributors, innovators that encourage you to engage with the diverse stakeholders groups. So such as we can say, let us talk example patients, healthcare providers, or advocacy organizations. They gather support from their project, they gather information, they gather different opinions from them. So this broad-based support can lead to more comprehensive and impactful in biomedical solutions. So that is all about. So in another case, let's give certain prototyping in healthcare services. As I said before, that the prototyping plays a crucial role in enhancing patient engagement by creating tangible representatives in healthcare solutions. By how? Let's take care of, for example, co-creations with these patients. So prototypes allow patients to actively participate in the design process. By how? it is just involving these patients, designers gain insights into their needs, preferences, and certain challenges. So co-creations that foster a sense of ownerships and empower patients more to involve in their solutions or problems. Okay, so by doing this, it is all about bring a good sense for us to understand about to involve these patients in a certain problems. We gather different informations, opinions from patients by involving these innovators or these professionals in a certain medical conditions or certain problems. And after there, we just visualizing their concepts. You know, prototypes transform abstract ideas into a visual or interactive forms, you know. So patient can better understand, they provide feedback on the proposed interventions, you know. So visual prototypes bridge the gap between technical jargons or technical professionals and the patient-friendly language, you know that. So that is all about. And about another issue, it is all about using of this interactive testing and refinement. No, prototyping enable this interactive cycles to testing and refinement. Designers create low fidelity prototypings, like it can be about paper sketches or high fidelity ones, interactive applications, whatever the case. But patients' feedbacks, informed adjustment, ensuring solutions aligns with their needs. Empathy and the empowerment about this. So prototypes evokes about empathy by making patients active participants. You know, patients see that their inputs matter, leading to increased trust and engagement. By how? You know, feelings here about understood, empower patients to take charge of their health. And remember, patients' engagement is all about collaboration, communication, and tailoring solutions to individual needs. So I can give case study about quadratic findings in action. Let's consider about hypothetical case where a team researchers developing a low-cost portable diagnostic device for early detection of infectious disease. During the prototyping stage, they seek maybe findings through quadratic findings in certain platforms. This project supports from a diverse group of stakeholders, including this local healthcare. You know, they just provide these issues of diagnostic devices in order to detect about the infectious disease. It can be about MRDT for the diagnosis of this malaria. We check urinary strips for diagnosis of different parameters in urinalysis such as glucose, leukocytes to detect for urinary tract infections. Or maybe it can be about the blood pressure machines to detect about higher blood pressure or hypertension, whatever the case, or RBG to detect for random blood glucose, to detect for diabetes mellitus. So they just gather about those informations to a certain community in order to come up with a wider range of knowledge to innovate about something, to come up with a good solutions about the community. So this project, you know, garner support from diverse group of stakeholders, local healthcare providers, patients, and these public health organizations. So we must involve firstly, let's say care providers, we must involve patients, and we must involve about public health organizations. So for bringing them all, we come up with a one solutions. You know, although individual contributions are relatively small, in quadratic mechanism can amplify more the impact, resulting into significant financial support for the project. That's why we always invite these donors to support about a certain project. With this, can increase adequate funding secured. No, the research team can proceed with the developing and testing about the prototypes. So conducting 3D, making trials, making about this researching. So refining this using device based on the feedback. So they come up with a broad-based support. it is fosters about these collaborations with the healthcare providers who offer about insight into practical implementations and potential challenges. Ultimately, I can say this: these projects progress rapidly through the prototyping stage, leading to a viable diagnostic tools that address about a critical public need. Conclusion, I can say quadratic fundings, represent a paradigm shift into the way of resources that are allocated to biomedical innovation ecosystem. So democratizing access findings encourage collaborations and efficient allocating resources based on communities' preferences or needs. Quadratic fundings have the potential to accelerate the prototyping stage of biomedical knowledge translation. As we say that biomedical field continues to involve embracing about the innovators ideas. So, quadratic funding can pave the way for more inclusive, brings about impactful and equitable advancement in healthcare system. So incorporating quadratic fundings into the prototyping stage, not only to enhance about this is development for biomedical innovations, but also to empower about a diverse range of the stakeholders to contribute and benefit from this advancement. So as we move forward, we have the summit on December. So we are going to explore and adopt funding models, different funding models. Partings to the crucial in driving about for the importance of next generations in biomedical breakthrough. So that is all about the prototyping in biomedical knowledge translation. In this part, we always customize about this care planning, buildings about relationship and communication, and involve decision making. So coordinating about this care across any team translations. Thank you. Thank you, Grace.
Thank you so much. Thank you so much, Damen, for that nice presentation. Hope now I'm audible enough. Thank you. So at this time, I want to welcome Dr. Macharia to give us more additional points about our topic for today. So welcome, Dr. Macharia.
All right, thank you. Thank you so much, Grace, and thank you, Damian, for that detailed explanation of the prototyping stage in biomedical research commercialization process. So, or translation process. Now, prototyping is when it becomes really hot. I mean, it's like the kitchen you are now cooking, and the fire is very hot, it's ablaze. I say this because at the prototyping stage, a different funding method in a traditional system, the different funding mechanism comes in. We discussed last stages from exploration to externalization to formalization and ideation. Those are stages usually funded through grant mechanism by the US government, National Institutes of Health, the National Science Foundation, and such organizations. And this is within the academic establishment, like a university, through the lab. So it is that you could say that is little money, because it costs maybe $100,000, $200,000 for doing that research work, exploration work, exteriorization, formalization, which is standardization of the idea, the concept, and ideation, going to conferences and making presentation. That's small quantities of money. So the government is able to come in and intervene, but only in the United States, maybe Europe, places like England, United Kingdom, places like Germany, France, they have money for that, but nowhere in Africa. We don't have any governments in Africa participating in funding the initial stages of a product. That is giving money to students and professors and people in research organizations to develop the knowledge. So once the concept has gone beyond the ideation stage, it jumps into the prototype. Now that stage, the funding mechanism is very tricky because it relies on what's called capital markets. You have to form capital. So you had an idea and you formalize it. So you come to ideation stage. You have the initial idea still on paper. How are you going to get money to fund that? So this is happening on the side of the industry. Now it has left the university, has left these gilded halls in the universities and colleges and labs and research labs. It has gone on the other side. So if you're still in a university, now you are subject to the market to go and raise money from the capital markets. How does that work? That's very hard. And then before you can even think about raising the money, it's how much money do you need to raise? And you determine how much money you need to raise after you have created the concept paper, the concept paper of your idea, which you'd call it like a business plan or white paper. You have done your calculations and seeing how much market are you going to reach with your product. Supposing it's like a biomedical diagnostic device, like for testing malaria, what you say. So you want, first of all, understand what's the total available market for this device. How many people are likely to use it? How many laboratories are there in this world if you capture the entire market? So then, of course, you're not going to be able to capture the entire market at once. It might take you 10, 20 years to distribute the entire market. So, but then you have total available market. This device is going to be available for 1 billion people, to 1 billion people. So you could say the total available market is 2 billion. But that market may not be accessible to you where you are. Say, for example, you're in Kenyatta University. How are you going to get 1 billion people from Kenyatta? You have to start at home because charity begins at home. So then from there, there's total accessible market. The ones you can access. Maybe you can say, because I'm in KU, I am going first of all to capture the KU hospital. Then you see how much money would that be by putting that device in that hospital, the TRH in KU. And then you say that is just too little. You can say I can capture KU, Nairobi Hospital, Kenyatta Hospital, and all those hospitals in the great Nairobi area or in the Nairobi County. And then you can start seeing that maybe then we are talking about access to 11 million people. Okay. So it's from that numbers you now calculate that if you are going to sell the device to serve 11 million people, how much money would be making in 1 year? Remember, so you can now start having idea about financial projections. So if you're going to sell the device to serve 1 million people, and maybe how many of those people are likely to consume service from that device? You see, it's about 15%. How do I get 15%? You pull it out of the blue, but you know that 15% of any population is going to consume healthcare at any one time. So 15% of 10 million people is about 150,000. Is it something like that? 150,000 or 1.5? Is it 1.5 million? That's 15%. I think it's 1.5 million in a year. Yeah, 1.5 million people. Okay, so if you say that, then you can say, if I am going to be charging just $1 per person who comes to consume the service, then I will be raising 1.5 million. I think it's maybe, what is 15% of 15% of 11 million? Hey Google, what is 15% of 11 million?
The answer is 1,650,000.
1,600,000. See, you can now say, I have a market of $1.6 million if I was going to be charging $1 per test, if all those people are going to consume services. But that's assuming that you are all alone. There's nobody else who is selling that device. Of course, You have to factor in competition. And then you say that to be very conservative, I'll get only 10% of that market. That's what's called market share. That my market share is only 10%. That's come to about $160,000 per year. That is significant. You see, that is now how you reason out. With your idea, you convert your idea into money. Now, in the traditional finance system, what people used to do is that you would create shares of common stock. Because imagine if you say the total market for this device is 1 billion people, you can actually you can register a company and authorize to issue 1 billion shares. Okay? If you're going to capture the entire market. Then you say that you only have Nairobi, which is 11 million. You say that I need to raise $11 million to get to Nairobi County, all of it. But then $11 million may not be so easy to raise. So you say, for me, I know that being very conservative, I would capture only 1.6 million. Okay. If 15% of the entire Nairobi County comes to consume my service. So I will I've authorized to issue 1 billion shares, but then I'm going to sell 1.6 million shares to raise money to finance the prototyping stage. Okay. So that's what's called capital markets. That's capital market formation. Have you understood the steps? First of all, you want to understand, calculate the market size, total available market, accessible market, and the market share you are likely to get, which means at the very end of the day, it's only $150,000. Now, if you show the market, I mean, people who investors, they are going to say, this man or this woman has really thought about this product. Remember, you started all the way in exploration, but then you're coming here to talk to the market and tell them, this now is what this product is likely to bring for me. So you can sell those shares. Why are people going to buy your shares? Is because when those shares eventually are listed in the New York Stock Exchange or in the NASDAQ, that's National Association Security Dealers Exchange, equivalent to the Nairobi Stock Exchange, they are going to raise in value and they shall make money. So people will give you money for that. Because you cannot get that money, you cannot sell that money ahead of time, you can go now to potential investors who are called angel investors or venture capitalists, and they put their money down and you can sell to them shares at a discounted price. So like instead of $1, you sell it at 90 cents or 50 cents of the dollar. So that is how people form capital for prototyping. Okay, it can be already an existing company, a going concern. For example, we go to already an existing company, say like it's a Nairobi hospital already. It has got they're in their research department, if they have one. Say like Kenyatta Hospital or maybe at TRH, Kenyatta Hospital has got a research department. They have found a new medical device and they want to fund its prototyping. Then they can create a company on the side, register the company, then they do the same procedure. Then they raise money from well-wishers, angel investors in exchange of shares. They give those people shares in the beginning, promise, but they sell those shares at a discounted price. So they can actually, they can be able to raise $1.6 million. And that money is going to go not to Kenyatta Hospital, but a separate company. A separate company, which is called a spin-off. It's on the side. So it doesn't show in Kenyatta National Hospital's bottom line. Okay. So this money is available for prototyping stage. And because that prototyping stage is supremely expensive, in biomedicine, particularly pharmaceutical science, it costs more than $1 billion to go through the prototyping stages, which I'm going to tell you briefly. So you can see, now that money is, if the investment in the beginning is coming from only a few people with deep pockets, investment bankers, or venture capitalists and the like. But then the people who are going to be using that, who are going to be using that technology, the biomedical device, maybe the patients, the community, or that 10 million people in Nairobi County itself. They don't get involved in that fundraising. And now that's what quadratic funding throws away. It just overthrows that, because with quadratic funding, now you are able to gain access to people without a lot of money, without having deep pockets. Just people can contribute small quantities of money in a community fundraising. So you get the entire community involved in buying the tokens, because the same thing, you would have issued shares, you create tokens, and then those tokens, you offer them to the people, anyone in the community, because they can afford 100 shillings, or 10,000 shillings, 20,000 shillings, not $1 million. And so you can get broad community participation. And what Damien has told us, this quadratic funding allows, first of all, the local community to participate in raising that large capital that is needed for prototyping. Okay, it has never been possible before. Now, what I've just described to you does not happen anywhere in Africa. That's why we don't do this biomedical translation. We don't understand something called capital formation, because capital formation, as I've told you many times in the past, is the reserve of the high priests of finance. Only those people who have got access to the holy of holies of finance, venture capitalists, investment bankers, and the like. But if you have an idea, you go to Nairobi Stock Exchange, where the holy of holies of capital. The people in Nairobi's capital markets, they do not comprehend. They have no clue what you're talking about. We talk about biomedical translation. That's cluelessness. And why are they clueless? This is because they're clueless. But now, the community itself, community members have a clue. Because community members have a clue that they need to get tested for malaria, for example. But the people who are working in Nairobi stock market itself is clueless. And what causes that cluelessness? It's, what would you say? What causes that? It's like they're in the shadow, they're in darkness. Is that darkness? So now quadratic funding helps to shed light in that darkness. And that darkness now gives people access to financing things that really matter to them. Okay, so it's going to be a very exciting period when these things happen, because we don't have to wait for medicines or biomedical devices to come from somewhere else. These biomedical devices are within us, and we're able to create them so long as we can organize the money. So why the funding is expensive for biomedical devices is because, as Damien told us, there's two things that are happening. You are developing the device, that's called the development, whether it's a drug or a device, and you're testing it at the same time. So you have a prototype, which is small. It is one object that you can now take to patient and start testing. But before you can take it to the patient, now the regulation requires that you go through 4 distinct stages, which they call clinical trials. Clinical trials. And the clinical trials are expensive. In the very beginning, you test it with animals. Say, so for example, if you created a diagnostic device, maybe an imaging device, you have, first of all, to take those images on an animal, like a dog, or a guinea pig, or a pig, or a cow. Why? Because this device can injure the living cells. And so you just want to find out whether it has got any effect. And if it's a drug that you're going to give people to destroy a disease like malaria, you first of all want to actually test it on the laboratory bench, before even you go to the animal studies, that you have to be on the bench and you're on a petri dish. Then you put that drug against that parasite and see whether it's killing the parasite. Those are laboratory bench studies. And those, once you get out of there, you go to do animal studies, live animals. And the live animals, like stage 1 of the clinical trials, you are testing the toxic dose. First of all, whether that thing has got any effect on a living human thing, not a human, a living thing, such an animal or a fish or chicken, that is to make sure that it is not going to be, it has no effect on life itself. So you just testing on the effect on life, okay? So if you establish that this thing, you can consume it and continue living, then the next thing you do, so you have to show, you have to publish those results. You take them to the lab and then you calculate and they say, wow, this thing was given to so many animals that none of them died. Good. Next up. Or say this thing was given to so many animals, but 20 of them died. Or maybe this thing was given to so many animals and 80 of them died. That's like 80%. That is bad. You can now stop the trial that it is killing 8. Then you can say, then you start testing what's called the toxic dose. At which point does this thing cause death? Or it's not just death that you're testing. There are other things that can be changing in the animal, in the living cells. It could be inducing multiplication of cells, or it could be reducing the multiplication of cells. So inducing is causing the cells to multiply many times, which can make them cancerous, or it can be killing cells, and even a particular type of cells. Okay, so that is the second thing you do is to check what toxic dose. What is the least amount of dose that you can give? And I think what is the dose that you can give that's going to cause the cells to get destroyed, the living cells to get destroyed? Toxic dose. So, and that toxic dose, or what is what is the minimum amount that you can give that is going to stop the growth of, say, like malaria parasite or multiplication? So these things, you still do them in animal studies. You're not doing them in human. And then you have to report that. That is a thing that you have to organize almost 6 months of trials like that, testing so many animals. And of course, maybe working in your own laboratory. But you cannot just test one animal and say that you've got the solution. You have to get quite a number of animals to test. That's why you need money, an effective lab, a clean environment. And then how can you say that it is what you're giving the animal that killed that animal, that's killing the cells? So you have to remove all the confounding factors. So really, this is a very tedious type of work, but you have to get through that because you're not going to get approved by the Food and Drug Administration in the United States. So then, after you have found the toxic dose, then you do another thing called the minimum effective dose. What's the least amount of drug you can give in order for you to produce an effect? For example, if you're making an X-ray for, a radiation machine for killing cancer cells, stuff like that. You say, what's the minimum amount of intensity that you can give to cause a positive effect? So that's another series of tests, and you can do it also in animal studies. You see, this time you are not even allowed to give your product to humans, because humans are protected. Only after you have established the toxic dose and the minimum effective dose, can you now come and do a small group of human studies, maybe a small quantities, like within one hospital or within 2 towns, like within, say, Kenyatta National Hospital. You can now give your molecule to maybe about 2,000 people. But that's 2,000, you have to recruit those patients one by one. It's very hard work, which you can now imagine already 1 year, 2 years have passed when you're doing all these tests, and then you're publishing all those results. You're not doing it secretly. You have to write it up and also submit all those results, the Food and Drug Administration, so you are allowed to go to the next stage. So you have to do small studies now to find what's called the false positive effect, where you can be giving your medication and it's showing false positive results. Those are people who are getting well without the effect of the medicine, but it's showing as if it's a medicine itself. Okay, so you have to eradicate that, find out what are the false positives, and then also the false negatives. All those people who have done epidemiology, they know what false positive and false negative means. So that you test it in a tiny population of about 2,000 people. And because you've been allowed to do human studies now by this time, this is about 1.5 years of testing. And then that's not enough. That is when you can now say in confidence that this thing is good in a very specific population. So for example, if you test your medications in, say, Kenyatta National Hospital or Nairobi Hospital, you are testing the local population there. But that doesn't mean the medication is going to be good in Kakamega Hospital. Okay, so you can now generalize the results. The results are local results. So in order to get out of there, now you have to do something called population studies, which is now the human studies, which is stage 4 of the clinical trials. Where now you can do a large study, multicentric studies. That's what they call them, multicentric studies, or the trials. When you ask teams in, say, other clinical teams, if you're doing infectious disease testing for your device, like diagnostic device for malaria, you can now recruit hospitals in Tanzania, in Uganda, people in the hospital laboratory so they can implement it. And then you have to go there and train them the protocol itself so that they can administer the protocol line by line by line by line. And so you have to employ people to go into those places who are going to be your clinical research coordinators, or CRCs, clinical research coordinators. And then they are going to be visiting the principal investigator in the group. So you have to identify a specific person there, and then you give him the work and the protocol. This person has to be qualified. He has to have knowledge of the lab. You have to train that person well enough. That means that they have to come through a training for about a week or so to learn what they're supposed to do in that protocol. So because they have to follow that protocol to the letter. And that is most of the studies that you find going on, like American organizations or clinical research organizations. They perform such studies, population studies, Kenyatta Hospital, and maybe Naivasha Hospital, and I don't know other hospitals in Africa, but that's the level where people are involved. But from fundraising perspective, none of the people in Africa, and even Kenya Medical Research Institute, KEMRI, is all those doctors working at KEMRI are doing those type of studies, like stage 4 studies. Can you imagine where we started? That's why we don't understand how to make our own stuff, because we only involved at very, very late stages of work. Now, so this is in human research. Now, people who are working in animal side, like International Livestock Research Institute in Kabete, may be involved in doing the animal studies in very early stages, because they are dealing with, they're dealing with animals and livestock. But most human studies that you find in Africa are usually stage 4 clinical trials, which is very, very late in the picture. And the meaning that they only get a visit from the clinical trial designer or the product designer, who really doesn't actually come there. They send a clinical trial coordinator who is an employee to come and teach our local professionals. So we are being taught by not the real designers. The real designers do not even leave Europe or United States. You can't see them because that work is very far beneath them. It's the work for going to train people in the village. It's like It's not in the village. I mean, for them, it's a village, but they want that these are population studies. Or if they have done stage 3 clinical trials, which requires much more attention, and they have been training these clinical trial coordinators all this time. So imagine now they have gone through this process for about 3 years. So the person who's been working there is well familiar with the protocol, and you need about 100 people who can be sent to different places to go and teach the people there, so they now can implement. So that process, now by the time you're getting, of course, after that you have to submit the document to the Centers for Disease Control, to FDA, to the Food and Drug Administration for regulatory approval. So that eventually, if you do that, the FDA is going to review all the data from all those stages, and they see how the data is aligning. And then if the device or the or the molecule is shown to be effective, clinically effective, effectiveness is that it can kill the parasites that you're talking about, or it can detect the parasite without killing the patient at all, and the patient is alive forever and ever, then you get approval. That process can take anywhere between 5 to 10 years, sometimes even 20. It's supremely expensive. So how do you raise money for that? Yeah, because people are not, they're not expecting to start earning revenue or dividends within 1 year, because you will not be able to sell that product for 10 years. Where does that patient come from? So it's called patient capital. The money that comes in has to be patient. You have to have people who are willing to wait that long. But then with the stock market, the promise that you're still doing studies and you're publishing, you're fighting clinical trials, gets people excited. And if it's a stock, the stock can start raising in value. But the same thing now, but that is only available to people with deep pockets, a few people. Now with quadratic funding, we can issue now the tokens themselves, and the tokens are in decentralized exchanges or even in centralized exchanges. And they are really independent of these stages of clinical trials. And the people who invested that kind of money for supporting that product have not put in too much money to worry about the number of years they're going to make. It's going to take. So that, because you can see this process can take 5 years, 10 years, even 20 years. Meanwhile, people who hold tokens, because they contributed money and they got tokens in exchange, those people will be trading those tokens in Binance, they'll be trading them in Uniswap, and that process is totally independent from the clinical trial process that we've just talked about. Okay, so eventually, when that FDA approves the use of that product, then it becomes available in the broad market, and everybody makes a lot of money, a windfall. So the price of the token will shoot up. But in the beginning, the price doesn't need to shoot up. They're holding the tokens and can be trading with it in the way they want. And so, as we have said before, tokens and quadratic funding disassociates or separates the funding itself from the product development. So people who are participating in product development can receive money for product development through quadratic funding without having, and then it can be using that money doing all those tests and trials, the testing, the iterative testing, without having to worry about the money. Okay, that's what's very, very revolutionary, and I believe that we want to understand this process very, very well. Because why is it not? Why is it not warning whether how does it disassociate? Is because quadratic funding involves many people contributing the money, and these are the people who have got specific interests in the product that's being developed. So long as people have got specific interest, even if it fails the clinical trials, they can do it again and change what the FDA says. You have to change this, you have to change this. Then there's funding available for them for making those changes. So eventually the product is going to come out because there's funding. But if there's no money, they will never see the product. So the only thing that lacks is not the products themselves, is the mechanism for funding. Now, ladies and gentlemen, we have that mechanism. So it is up to everybody here to understand that. That process has been a secret process, which is only reserved to the high priest of finance, who in the Holy of Holies, it's like the Holy Temple of King Solomon, which had that space where the only one priest could get in to pray for the sins and to perform ordinances, to ask for forgiveness for the entire population. Just one priest. Okay. Now we're talking about the entire population participating in praying for itself, for forgiveness of its sins, so it can get access to heaven. Okay. So that's the same thing that's happening. So as we said again before, is that the curtain of the temple has been split way down. People have got access to the Holy of Holies, such that they can participate in performing their own ordinances for their own salvation that will take them to the kingdom of God. So that is the story about a very complicated process, which has been engineered to make it that way, to make it out of the reach of the people, but quadratic funding makes it absolutely accessible to all of us, unless we do not want to do it. But as we said, we are not of those who draw back to perdition. We are not of those who draw back to perdition. We are of those who go to the Holy of Holies, and that's what we are doing. Okay. Well, that's it for now, ladies and gentlemen. Back to you, Grace.
Thank you so much, Dr. for that wonderful presentation, wonderful comments about quadratic funding in the prototyping stage, helping us get more understanding on what prototyping is. And thank you for that nice presentation. Thank you so much, everyone, for listening. So at this time, maybe if there's anyone with a comment or a question, you may maybe raise your hand. Because of time, I will be posting the attendance form on the chat on the message side. So kindly fill the attendance form. Let me welcome anyone with a comment. I have posted the attendance form on the chat box, so kindly fill that form. Because of time, since we are going to join the other meeting for African Women in DeFi, if there's no one with the question, I think we can adjourn the meeting. See you in the next meeting. Thank you.